So thank you so much for coming to my talk, the first talk of the meeting. My name is Corlett Wood. I'm an assistant professor at the University of Pennsylvania. And today I'm going to tell you about a project I did with a former postdoc in my lab, Emil Gluck-Thaler, studying multivariate divergence in wild microbes. So organisms are composed of suites of genetically integrated or correlated traits, like in this hypothetical example of a Galapagos finch, with beak length and beak depth being correlated. We typically represent genetic correlations as these elongated ellipses where the long axis of this ellipse is referred to as the major axis of genetic variation, or Gmax. And it's the multivariate combination of traits that contains the most genetic variation. Genetic correlations influence the response to selection because if you impose selection on a trait like beak length in this case, you'll see a correlated response to selection in beak depth. And something that you should notice right away is that the major axis of genetic variation biases evolution in the same direction as it, in the axis where there's the most trait variation. So in a landmark paper in 1996, Dolf Schluter termed or coined the phrase the genetic line of least resistance to capture this phenomenon of evolution along the major axis of genetic variation. And so in his landmark paper, he famously predicted that over long evolutionary timescales, genetic correlations would influence divergence between species and populations as well. So we made two predictions, at least that I'm going to talk about today. The first is that populations and species that are very recently diverged from a common ancestor should differ in a direction close to the direction of greatest genetic variation. And so if the ancestor is that ellipse in the lower left, you can see that the most recently diverged over short evolutionary time, we see divergence right aligned with Gmax. His second prediction was that this bias in the direction of evolution should diminish with time, or should get weaker over longer divergence times. So he tested these hypotheses in 5 vertebrate taxa in this initial paper, and he found support for both of them. And since this landmark paper, there have been tons of subsequent studies that have done something similar. I'm showing you only a representative sample right here. And so in the last 20 to 30 years, we've amassed a ton of examples of this phenomenon across a ton of different taxa. You can see examples from vertebrates, invertebrates, and even a plant example on here. But there's one group of organisms that's conspicuously absent or underrepresented in this literature, and that is microorganisms. So in contrast to macroorganisms, where we now have a several decade-long history of thinking about multivariate patterns of evolution, fungi and bacteria and other microorganisms are conspicuously underrepresented. And so Emil's and my goal in this study was to address 2 questions. First, to test whether genetic correlations are conserved across microbial lineages, and second, whether divergence occurs along the genetic line of least resistance in microbes. And so why might genetic correlations play a different role in microbial evolution? Well, there are potentially a number of reasons this might be the case, but I'm going to focus on 2, both related to differences in the mode of inheritance between microbes and macrobes. So first, asexual reproduction is a lot more prevalent in microorganisms than in macroorganisms, and there's some evidence in the literature that patterns of genetic correlation differ between sexual and asexual lineages. And second, horizontal gene transfer, or the movement or mobilization of DNA between lineages, is much more common in microorganisms, and this is even true for eukaryotic microorganisms like fungi. So I think the role of horizontal gene transfer in multivariate evolution is especially interesting because you could imagine that mobile genetic elements could potentially restructure genetic correlations fairly rapidly, or depending on the suite of traits that are mobilized, could even transport a population rapidly to a new region of phenotype space that's not aligned with this major axis of genetic correlation. Okay, so in this study, we focused on this group of microscopic fungi called dark septate endophytes. These are melanized ascomycete fungi. They're abundant in terrestrial ecosystems. They have both endophytic and saprotrophic life stages. When they're endophytes, they colonize the roots of vaccinium species like blueberry and cranberry. And as saprotrophs, they live on wood and then leafy debris. And we know that their interactions with their hosts vary from commensal to mutualistic to pathogenic. And so in this study, we sampled endophytes from 3 different study sites in Pennsylvania and Virginia. At each site, we collected blueberry roots from low bush blueberry. We cultured endophytes out of those roots on selective media, and then we identified them using ITS sequencing back in the lab. And so today I'm going to tell you about a group of 30 isolates 13 of them cluster into this yellow clade labeled clade D, which is a monophyletic group, and the other 17 cluster into this clade I'm going to refer to as PAC, which stands for the Phylocephala acephala species complex. So when we measure traits in microbes, we obviously need to think a little bit differently than we do typically in macroorganisms. What I'm going to focus on for the purposes of this study are microbial metabolic traits, where a metabolic trait refers to a microbe's growth on a different metabolite or a different substrate. And so in our study, the genetic correlations I'm going to be talking about are genetic correlations between growth on different metabolites or different substrates. In this study, when we're estimating genetic correlations, or specifically genetic variance covariance, or G matrices, we're doing it based on isolate means, where each mean is an average of 3 different replicates. Okay, so we estimated or measured microbial metabolic traits in 2 separate experiments. The first I'm going to refer to as the metabolite experiment. And the goal of this experiment was to measure microbial responses to a bunch of different host metabolites that we know are produced by Vaccinium species. And so here we used a panel of 6 phenolic compounds, which are putatively growth inhibiting, and 5 carbohydrates, which are putatively growth promoting. In this second experiment, which I'm going to refer to as the plant extract experiment, our goal was to capture microbial responses to realistic multivariate biochemical environments, or how they might actually respond to growing on a host. And so we did this with extracts from 5 different plant tissues, 2 from the primary host, either the leaves or the roots of low-bush blueberry, and 3 from other sympatric hosts, the leaves of pine, oak, and maple from the same study site. So for each of these experiments, we streaked our isolates onto plates that contained each substrate, and then we measured growth as colony diameter on each of those plates. So first we asked how metabolic traits have diverged between endophyte lineages. And so here in this graph, I'm showing you the different metabolites in our metabolite experiment on the x-axis, and you should be able to see the control plates on the far left. And so what we see is that the PAC isolates from this green group grew faster than clade D isolates across nearly all metabolites. With the exception of caffeic acid right here, essentially caffeine, which abolished growth differences between our groups. If we then lump together all phenolics and all carbohydrates, you can see that growth differences between our lineages were significantly greater on carbohydrates than they were on phenolics. And something that was counter to what we expected is that phenolics were not growth inhibitory in this particular experiment, which was surprising to us because they're commonly produced secondary metabolites that plants are typically using to suppress the growth of microorganisms. So we then repeated this analysis for the plant extract experiment, and so what I'm showing you are now the different plant extracts on the x-axis, and we see a qualitatively similar pattern where isolates grew faster from PAC than from clade D. We also saw that growth was slowest on Vaccinium, which is our native or primary host, which was surprising. We found that all of our metabolic traits were generally positively correlated, so I'm showing you this just briefly in principal component space for the 2 different isolates in the metabolite experiment. It's a little hard to see on these graphs, but the first principal component explains the vast majority of variation among our isolates, and all of our metabolite traits load positively onto that principal component, indicating positive correlations among them. And we see a qualitatively similar pattern for our plant extract experiment as well. So what we then wanted to ask was whether these patterns of genetic correlation between these metabolite traits are conserved between our 2 lineages. And so we used 3 different metrics to test this. First, we compared the total amount of genetic variation between lineages. Second, the magnitude or strength of genetic correlations. And third, the orientation of genetic correlations, or this is another way to think about this is the direction of the major axis of genetic variation, or Gmax. So in each case, what we did is we compared the observed difference, which I'm going to show in that dashed vertical line, against a null expectation generated by permuting isolates among lineages. And so what we see is that there's a significant difference between our lineages in the total amount of genetic variation across these traits in the metabolite experiment. But we see no significant difference in the magnitude of genetic correlations. And we actually see when we look at the direction of Gmax that it's more similar between the lineages than we would expect under the null. And so we see some evidence that G or genetic variance covariance matrices differ a little between lineages, but mostly that's driven by total amount of genetic variation. We then performed the same analyses for the plant extract experiment, and we see no significant difference across any of these 3 metrics. And so generally, we're seeing pretty strong evidence that patterns of genetic correlation are broadly conserved between these two endophyte lineages. So finally, we wanted to ask whether divergence between these lineages occurred along this genetic line of least resistance, as Schluter predicted. And so to do this, we measured Schluter's theta, which is the angle between this major axis of genetic variation, or Gmax, and the direction of divergence. And so if this angle is 0, like in this top example there, that indicates that divergence was perfectly aligned with the major axis of genetic variation. This is what Schluter expected. If the angle is 90 degrees, this indicates that divergence was perfectly orthogonal to that axis of major genetic variation. This is not what Schluter expected to see. And to our surprise, when we looked at the metabolite experiment, we see that divergence between lineages was nearly orthogonal to this major axis of genetic variation. I'm plotting the two lineages in principal component space after removing average differences in growth between them. And what you should be able to see is that the major axis of genetic variation within lineages is aligned in this direction, while divergence between them is orthogonal. And we see something similar when we look at the plant extract experiment too. Although my slides are not advancing. There we go. And so we see that again when we look at the plant extract experiment, the divergence is orthogonal to this major axis of genetic variation. Okay, so why might we see divergence that's not aligned with Gmax, or this genetic line of least resistance in this particular group of fungal endophytes? We're certainly not the first study to see this. I've listed several others on the bottom of the slide that have found cases where divergence is not aligned with the genetic line of least resistance. And they've floated a couple of hypotheses. One obvious one is that we may have omitted a genetically variable trait that's under strong selection. So our G matrices don't actually reflect the relevant patterns of genetic constraint in this system. That's one possibility. A second related possibility is that our contemporary measurements of genetic correlations in these populations don't reflect the historic patterns of constraint that actually drove or constrained divergence. Although I think that's relatively unlikely here, just because there aren't major differences between these lineages in the structure of genetic correlations. A third possibility is that these endophyte lineages are more distantly related than we think that they are. And as you might remember, Schluter predicted that the importance of this genetic line of least resistance should decay over evolutionary time. But again, sequence similarity suggests that these things are in the same genus. They're fairly close relatives. So we think this is not a super likely explanation either. And so that leaves us with a fourth possibility, which is that horizontal gene transfer is contributing to divergence in this group, and it's freeing these endophytes from the constraints of the genetic line of least resistance. So this, right now, is just a hypothesis, but I think because of the dominance of macroorganisms in this literature, it's untested and a really interesting possibility. So I just want to end with a plug for using wild microbes as systems for quantitative genetics and evolutionary genetics. They certainly have their unique challenges. One of them you might be thinking of is defining a species in microbes. So distinguishing between intra and interspecific genetic variation microbes is uniquely challenging. Another is that targeted sampling of a focal clade in the field is difficult in microbes because you don't know what you have until you either culture it or sequence it back in the lab. But there are also unique possibilities, and I think a major one is the possibility of high-throughput phenotyping of these critters using something like high-throughput plate assays that measure growth on different metabolites. And so with that, I'd like to acknowledge everybody who contributed to this study and take any questions if I have a little bit of time. Thank you.
Okay, tough act to follow. I never really thought about horizontal gene transfer and genetic correlations, but I'm excited to be here anyway. I'm Jake Botello. I'm a PhD candidate at Auburn University in Dr. Matthew Wolak's lab, and I'm excited to talk to you guys today a little bit about how those genetic correlations might interact to influence the evolution of sexual size dimorphism. So sexual size dimorphism is this widespread phenomenon that we can observe in sexually reproducing organisms of many different kinds, ranging from insects, mammals, birds, frogs, even plants in some cases. And a keen observer might notice that it's not always the males who are bigger. We often have females who are bigger than males, depending on the circumstance. And that leads me to wonder and ask, why do we see these differences in sex? Why do we see sexual dimorphism? And the answer is generally that we see the sexes responding to highly different needs. For example, if you're a female, you might be under strong fecundity selection to have bigger clutch sizes, lay more eggs, have more offspring. As a male, you might be under selection to be more competitive in male-male competition to try and secure a mate. So how do we actually get there? How do we get to this point where you've got this bimodal distribution of phenotypes specific to the sexes? Well, it makes us think about the evolution of sexual size dimorphism. How do we arrive at sexual size dimorphism? How can the sexes grow to differ so much? Well, the short answer is they often don't differ that much for a while. So what's good for one sex may not be good for another sex. We run into something called intra-locus sexual conflict, and this happens when a group of alleles controls a trait, but the sexes have different phenotypic optima, or fitness optima, for that trait. And this little elegant graphic by Cox and Calsby kind of illustrates the point well. If you look at panel A, you can see an example of no conflict where the fitness optimum, represented by that dotted line, is there in the middle, and it's perfectly aligned with the sort of phenotypic mean in the population. And if you look down at the bottom right, you can see fully resolved conflict, where sexual dimorphism has taken root, and you've got two distinct phenotypes for each sex. But in section B, or panel B, you can kind of see what it looks like when there are pressures for each sex to move in a different direction, but the sexes are constrained by one another. So it's a process. This takes some time. Some commonly proposed genetic mechanisms that help to explain how we end up with sexual dimorphism are the development of sex chromosomes, sex linkage. Sometimes we think about sex-specific non-additive effects where the autosomes interact differently across loci depending on the sex in which they are expressed. And I want to remind everybody that the selection that's occurring here is selecting for phenotypes. So even though we're thinking about genetic mechanisms, this is still occurring in terms of phenotypes. So we want to get to the bottom of whether or not there's some genetic relationship between these traits, or the genetic architecture that's influencing these traits, that might influence how sexual size dimorphism evolves. So rewinding back for a second to intralocus sexual conflict, this typically, when it's studied, takes a multivariate view. It looks at more than one trait at a time. So maybe something like body size and longevity, or metabolic rate and length, things like that. But investigations of sexual size dimorphism often focus on just a single trait. They're a story of one trait, and that's size. Or maybe some proxies for size, maybe like SVL, full contact. Maybe like SVL or mass, but it's all centered around size. So what if there's another trait involved in the evolution of sexual size dimorphism? Something that's not directly related to size the way we think about size. So I borrowed this cool graphic from Krishop Lab that shows male and female fitness optima, sort of represented by clouds, and imagine those sort of coming out at you as little mountains in the sky. And we typically see this represented where size might be on the x-axis and another trait might be on the y, and the expectation is that there are major differences along that x-axis between males and females, but nothing really major with respect to what's on the y. But we might actually be looking at something more like this in the wild, whereas one trait changes, the other trait changes as well, even in terms of genetics. So I want to think about selection on correlated traits for just a minute. So I want to take a real-world example to illustrate this, Callicebruchus maculatus, which is an enormously photogenic beetle. This is the first beetle that ever let me photograph him. This is Gus. We did a whole photo shoot. You can see me after for photos from the reel. So Callicebruchus maculatus is a bruchid beetle. These guys are invasive. They're pests. They love beans and peas. They consume all the resources they're ever going to have as a larva. So before they pupate and become adults, they've eaten everything they're likely to eat. Their larval development period takes about 25 days, and then they emerge as a fully formed, sexually mature adult. They have a short longevity. It's between 1 and 3 weeks. It varies dramatically across males and females. Males tend to live about a week, females about 3 weeks, or up to 3 weeks. And they're polygynandrous, so males and females all have multiple mates in a lifetime. So in this species, development time and mass at emergence are highly phenotypically correlated traits. So could some shared genes influence the evolution of sexual size dimorphism with respect to development time and mass at emergence? We know that these are highly phenotypically correlated. The longer a beetle tends to spend in development, the larger it tends to be at adulthood, right? So we know there's a phenotypic relationship. Could there be shared genetic architecture that maybe influences how size evolves in the sexes? We ran a quantitative genetic analysis to figure out how much variance was there for these traits. But before we dive too deep into the experimental design, I want to point out that we typically think about variance partitioning as being with respect to a whole population of individuals. And in fact, this can actually be split differently across the sexes because there could be different sources of additive genetic variance in males and females. For example, the pie charts here, I think on your left, you can see that males, of course, have, in some cases, Y-linked sources of additive genetic variance, whereas females do not. So you can see different types of additive genetic variance, or at least different sources. All right, so in terms of our actual setup here, we ran a breeding design, a double first cousin design according to Fairbairn and Roth. We extended that by another 6 generations by mating distantly, or at least not closely related individuals randomly, up to about 9 generations. We measured 15,000 individuals, and you're probably thinking, Jake, why only 15? Why so few? Well, it's okay that you're not impressed because the real number is 25,000 individuals. So many beetles, so many personalities. For all these beetles, we measure their development time. So that's the point from where they're on the bean as an egg until they pupate fully. And then we took the mass of those beetles in their first moments of adulthood, the day they emerge. So we really cover the life cycle of the beetle here. If you'd like to hear more about how we assayed these beetles for fitness and selection analyses, you can go to my colleague Kendall Skelton's talk on Tuesday at 11:45, part 2 of this. So what did we find? Well, I'm going to break this down from simpler to more complex. So let's start with some simple narrow sense heritabilities. And just a quick reminder, this is autosomal VA plus sex-linked VA over phenotypic variance, just really simple narrow sense heritability. So we see huge differences by trait. So you can see there's much more heritability in mass at emergence than in development time. If you can't see, that's 61% for females, about 70 for males, and only about 0.03 and 0.05 for females and males in development time, respectively. So the implication here is there's much more variation for one trait than the other. But here we've got a G matrix on the diagonal, top left down to the bottom right. What you're seeing is mean standardized variances. So this gives you a sense of how much variation is present in each of these traits. And then on the off diagonals, you can see correlations between those traits. So these are not all the same things. You can see in orange or brighter colors are stronger correlations or higher variances. Darker colors are lower. So you can see there's much, much, much more variation in mass, like we said. And we also see some strong cross-sex correlations. You might see those there, male and female mass and male and female juvenile development time. So let's break this out even further. And now we've got autosomal variants here on the far left. So this is only additive effects from the autosomes. X-linked effects in the middle and Y-linked effects, that's specific to males on the right. There are some gaps there where there are NAs in applicable values or things that just actually got estimated at 0. And if you look carefully, you can see there is still the same sort of general higher variance in mass than in development time. But you might notice in the top left of the left chunk there, the left G matrix, there's much more variance in the autosomes in females for mass than in males, but you can kind of see it adds up if you look at the male values for X-linked and Y-linked variance in that trait. So what does this all actually mean? Well, let's apply selection and let's see what happens when we try to expand our sexual dimorphism given this G matrix. So we started out with 37.7% sexual dimorphism index. This is just a simple calculation, basically the bigger sex over the smaller sex, minus 1 times 100. We applied multivariate selection according to the Landy 1980. This is directional selection for just one generation. It's sexually antagonistic selection, so they're opposite. And we applied 0.25. This is an arbitrary number, arbitrary selection gradient, but just to show you it's even in both directions. What did we find? Well, we compared selection response in our G matrix, in a G matrix that constrains all cross-sex correlations to be 0, and in one that constrains all cross-trait correlations to be 0. And what we found is our matrix, the real one, grows by about 0.5% in terms of its sexual dimorphism index. Okay, not too shabby. The no cross-sex correlation G matrix grew by 2%, so 4 times as much. And without trait correlations, we see only 0.5% growth again, which kind of suggests that maybe that's not a big part of the story. If you are clever, you might have thought, well, what about the cross-sex, cross-trait correlations, like female juvenile development time on male mass at emergence? How might those interact? Turns out it's the same as if you constrain the cross-sex correlation, which really just reinforces, it seems like the big story here is this cross-sex genetic trait correlation, or genetic correlation. Okay, so the take-home here is evolution of sexual size dimorphism is usually a story of just size. In our case, we took a multivariate, multi-trait view, looking at size and development time and how they might be linked. Although we didn't really find a strong genetic relationship between those traits, we did find that the cross-sex genetic correlation does constrain the evolution of sexual size dimorphism in our beetles. And with that, I'd love to thank the Wolak Lab, everybody who contributed, Dr. Charles Fox, who seeded our seed beetles, NSF for funding this. And with that, I'm going to leave you guys with what you all came here to see, the smolder. Thank you. I'm not sure how much time I have, but I'm happy to take questions for upwards of 2 minutes. Yes, sir.
Cool talk. I just have a question for you. B matrices between did you check the asymmetry of these matrices?
Did I check the estimates?
Yeah,
Oh, the asymmetries. I did not. That's a good idea. I should do that. Mm-hmm.
Was there much Y-linked variance in size and development time?
There was, I can show you if I back it up here. So a little bit. We're talking about Y linkage strictly in males. None of the Y linkage was particularly gigantic. It was enough to sort of explain the offset of males for the autosomes, but it wasn't anything to write home about. Yeah.
Imply your population doesn't have that Y-linked size thing that Alina Emmerman's group have discovered.
Yeah, I don't know. Anybody else? Okay, I'll turn it over to the next person. Thank you very much.
Hi, everyone. I'm Sarah Yakimowski from Queen's University, just a few hours away from Montreal here. Today, in the session, we're focusing on quantitative traits, polygenic traits that deviate from the simple non-Mendelian expectations. And there are many genetic factors that can cause some of these deviations from Mendelian expectations. We've heard about some already today in this session. So gene drive, which can create distortions in segregation ratios, transposable elements, the movement of genes through the genome, and epigenetic inheritance, where the expression of a gene is affected by processes other than the gene sequence. So today I'm going to be telling you about a quantitative genomic trait where we're interested in patterns of inheritance and are finding patterns that deviate from our expectations as well. So the study system we have been working with is Amaranthus palmeri. This is an agricultural weed that is expanding its range. It's one of the top 10 crop weeds in the United States. It is wind-pollinated and dioecious, so it has separate sexes. And it is resistant to at least 8 different chemical classes of herbicide, including glyphosate. So this weed is very problematic for our producers, and as an evolutionary biologist, it's also a prime candidate for understanding rapid evolution. Resistance to glyphosate in the system is conferred by a gene copy number mechanism. Let's see. Does my cursor work? No. Okay. So this gene copy number mechanism is part of a fundamental plant pathway, the shikimate pathway, and when there are duplications of the EPSPS gene, more gene product is produced, and glyphosate binds to, glyphosate binds to this enzyme, and if there is enough of this enzymatic product left over, this fundamental pathway can continue to function and render plants effectively resistant to this herbicide. Some of our previous work has shown that this copy number variation explains at least 60% of the phenotypic variation in resistance among populations. And across all individuals, there is a strong threshold relationship between copy number of this gene and phenotypic resistance. So there's a huge increase in resistance from an initial duplication up to about 15 copies, and then a much more shallower increase in resistance as copy number increases up to over 160 copies, which can be detected in this gene copy system. In addition to all of these gene copies, they are also found to be extrachromosomal. So they occur on structures that are tethered to the outside of the chromosome. And the shape of these are polymorphic. So about 50% of them, most of them are circular. But they can also be linear or irregularly shaped. And so this raises a lot of fundamental questions about the stability of these structures and how stable or unstable they are through the processes of mitosis and meiosis through reproduction and therefore ultimately raising question of how their dynamics in evolution. So this is what my student, Lisa Hahn, was interested in for her master's thesis. Unfortunately, Lisa couldn't be here with us at the conference. She's currently in the field. But Lisa's research focused on understanding what is the relation between the parent and offspring copy number in this species, and across the dynamic range of this copy number variation, are there differences in this pattern between parent and offspring inheritance? So to do this work, Lisa performed genotype-informed control crosses. So she grew lots of juvenile plants, extracted their DNA, and used digital droplet PCR to quantify the number of the EPSPS gene relative to a reference gene, ALS. And then with this copy number variation information in hand, she created controlled crosses using pollination bags, grew the plants to maturity and matured the seed, and then ultimately grew the F1 progeny and then estimated the copy number of those F1 progeny. So when Lisa first did this large experiment and she brought to me this first large data set, I thought, okay, wow, there is a lot of variation here. So each box plot here is an individual cross. And they are ordered from lowest to highest mean parental copy number. And initially, to be honest with you, I was concerned about the role of contamination in this wind-pollinated plant, given all of this variation. But we went on to analyze Lisa's data, and Lisa had put a lot of effort into doing high-quality crosses. Ultimately, Lisa detected a moderate estimate of heritability using a linear model, but Lisa also noticed some nonlinear components to the data and found a significant quadratic relation in the data as well. We went on to investigate this potential nonlinearity of the data through also a threshold change point analysis and did detect a significant change point, suggesting that there's much higher heritability from low to sort of moderate copy number, and then a much lower heritability from moderate to high parental copy number. Also, when we visualize the pattern of directionality, so the difference in the mean F1 copy number relative to its parent, we saw that from low to moderate parent copy numbers, we consistently see the maintenance or increase in copy number. However, all of the crosses with the highest copy number consistently exhibit a decline in copy number. So when I saw this analysis, I started to think, okay, contamination probably didn't create this pattern. However, we were still concerned, and so we went on to create 9 more crosses that were extremely isolated. We had only a single cross in the greenhouse at a time and no other plants growing of divergent copy numbers to get some really targeted data where we could rule out contamination. So when we add these 9 really isolated crosses to our data, overall the pattern looks very similar to the large initial data set. And overall we find that both the quadratic and threshold fits to this data are a better fit than the linear, supporting this sort of nonlinear relationship between parent and offspring copy number. And again, when we add these isolated crosses to our investigation of directionality, Lisa was able to add some single copy number crosses as a control. So you can see the parent and offspring were able to maintain that single copy number in those crosses. And again, we are able to see that from low to moderate copy number, there is a maintenance or increase in copy number, and a high parental copy number cross again confirmed this decline in copy number. So this is consistent with there being some kind of instability or genomic constraint on the maintenance of this high copy number. Also, if we zoom out again to the big monster data set, I'm not going to have you look at this for more than a second, but I am just going to zoom in on a few of these isolated crosses, the ones with shading here, where you can really see that there are F1 progeny, so the parentals are the red and the blue dots. And so you can see there are F1 progeny being formed that exhibit higher copy number than either of the parents did, in this case too. And in some crosses in particular, a lot of F1 progeny are exhibiting an increase in copy number relative to either of their parents. And so this brings to the forefront a hypothesis that has come from Mullen et al. Based on genomic sequences and the possibility of a self-replication mechanism. And it has also been proposed that this self-replication mechanism might be further instigated by glyphosate stress itself. Now, the populations we have been studying likely or almost certainly have a history of glyphosate use, but we did not use any glyphosate or apply any glyphosate stress during our experiments. We used seed families that were collected from the field, but grown in benign greenhouse conditions. So one of our next steps is going to be to impose that glyphosate stress and to further quantify how these dynamics are affected by that stress. So in conclusion, the EPSPS gene copy number heritability varies across its dynamic range. Matings between low to moderate copy number plants yield stable or increasing copy number, and matings between the highest copy number individuals result in a very consistent decline in copy number. And overall, this does suggest that copy number resistance gene may be approaching its upper evolutionary limits due to this constraint. And interestingly, based on our surveys of, here's just a couple of examples from populations in Illinois that are representative of what many populations look like. The highest copy number individuals in our studies have only ever been detected at low frequencies. So this is also consistent with this genomic constraint. I also just wanted to plug my students, Tammy and Charlotte, who are sitting there in the second row. Charlotte is going to be talking about the evolutionary origins of this copy number variation using a herbarium study tomorrow, on Sunday. And Tammy is going to be talking about some modeling work that has begun to understand the evolutionary dynamics of this resistance mechanism. And finally, I just want to thank all of my students, collaborators, and funders. And I have a couple minutes for questions. Thank you. Yeah, that's interesting. So actually, we do find 50 copies as being like a very common mean resistance in populations, and I do also see some evidence of stabilizing selection around that mean as well. So it's kind of fitting together. Hi.
Great talk.
Thank you.
Do you have any hypotheses for what the mechanism might be underlying the loss of the ZPS-based copy number? Is it like issues for actually like physically tethering the chromosome when there's so many, or is it possible this actually costs that you're just not seeing offspring that have a super high copy number in the
Yeah, I don't know. I think the way we've been thinking about it is that there are likely sort of physical constraints and damage. You're probably familiar with the Ku et al. Paper, so they do also find variation at different mitotic stages, and I didn't have time to show it, but we've also quantified the variation in different tissue samples, and we find that intra-individual copy number also increases with high copy number, consistent with that. So that's one of the hypotheses, but yeah, we're not at that level. Oh, thank you.
Hi everyone, I'm Euan. I'm a fourth year PhD student at the University of Groningen in the Netherlands, and what I'm really interested in is if evolutionary biology can be used to inform our understanding of human health and disease. And for my final PhD chapter, I tried to look at if the evidence for a genetic basis for how life history traits shape breast cancer risk in women. So breast cancer is this really devastating disease. It's one of the leading causes of death amongst cancer types. And this is quite remarkable given it's almost exclusively found in women. And although factors such as a high BMI and older age are really leading breast cancer risk factors, it's also been shown across a array of studies that life history traits play a role. Particularly early ages at menarche or development, later ages at first childbirth, and overall fewer numbers of children increase a woman's breast cancer risk. And this is thought to be due to a sort of evolutionary mismatch. Here I'm showing the birth rate per 1,000 across the Netherlands, and in the Netherlands, as with many countries, birth rate has really quite dramatically fallen over the past 80 or so years. And what this has meant is that women are experiencing many more menstrual cycles than our cancer suppression mechanisms have evolved to handle, and as a result, a much higher exposure to estrogen throughout their lifetime, which has proliferative effects on mammary cells. Although these phenotypic observations are fairly well established, we also know that these traits are to some small degree, influenced by genetic differences amongst individuals. It's been shown in the UK Biobank that around 20% of breast cancer risk can be explained by genetic factors. So this kind of begs the question for me is that, so are these observed associations, are these due to genetic differences at all, or are they simply plasticity? So some studies have tried to look at this using GWAS studies, and GWAS studies are quite good at finding associations between loci of very high influence. Yeah? But with traits such as these life history traits and also breast cancer risk, the traits are really quantitative traits. Here's a GWAS from breast cancer in the Japanese population, and what you see is really only a handful of traits with a really high association. A handful of loci with a very high association with breast cancer risk. So to really try and answer this question, I would say that taking a more quantitative genetics approach is necessary. And this could be really useful because it'll kind of help inform individuals of very high risk in the era of so-called precision medicine, and also trying to maybe answer more fundamental questions about why these breast cancer risk factors exist. Is it because they're also genes that are also important in regulating human life history traits? And I think the other nice thing about this approach is that actually just it can enable us to potentially rule out the importance of genetic factors here, which again would be useful. Okay, so to try and do the study, I turned to this Dutch biobank called Lifelines. It covers around 10% of the population in the northern provinces of the Netherlands. It's a multi-generational cohort study, so it contains health records, questionnaire data, and importantly, genomic data across the individuals. And to help us with this project, we reached out to our collaborators at the University of Groningen's Medical Center, just to bring on their really expertise in the epidemiology of breast cancer, as well as using this biobank data. So we then took our biobank data, and we linked it to the National Pathology database in the Netherlands. So the Netherlands is quite unique in that it's had basically national coverage of highly detailed pathology records from around 1991 entirely. And what this means is that with relatively high precision, we're able to determine the onset of breast cancer for individuals within this biobank, and also information around the nature and grade of the tumours. Upon linking this data, we had a prevalence of around 4%, so around 3,600 first tumour breast cancer records from 85,000 women in the biobank. Here I'm just showing the distribution of the age at diagnosis of breast cancer, and what you see is really quite typical in that it really shows this peak in midlife. And yeah. We quantified our life history traits we were interested in through the questionnaire data. So women were specifically asked, what age did you start menstrual cycling, the ages and number of children people have had. You'd notice that the sample sizes are a bit lower for the age at first birth and number of children. That's just because we restrict it to individuals that had mostly finished reproducing age. We then took our phenotypic data and brought in the genomic data. So we used this genomic data to build a genomic relatedness matrix, where essentially you compare every woman in the population and count the number of the proportion of single nucleotide polymorphisms in common. This was used using this Infinium global screening array, which gives you 500,000 quality-controlled loci across 36,000 individuals within the population. We then performed a genomic restricted maximum likelihood analysis using the software GCTA. So essentially we set up 3 bivariate case control studies with our breast cancer diagnosis, yes or no, and each of the life history traits. And then leveraged this variation among relatedness amongst individuals to estimate the heritability, or the SNP heritability, and genetic correlations of each of our traits in each of the models. This reduced our sample size a little, but still gives 18,000 women, 700 of which contracted breast cancer. Okay, so I'm going to move on to the results now. I'm just going to show you the heritability estimates of the breast cancer and the breast cancer risk and the life history traits, and then the genetic correlation between them. We showed that our life history traits we selected are to some small degree, influenced by these genetic differences amongst individuals from around 20 to 35%. Our breast cancer risk, there's also some degree of heritability there, but you'll see the 95% confidence intervals, the error bars, overlap 0, so we're not finding any significant heritability there. And when you try to estimate the genetic correlations with these data, the confidence intervals really become very wide. There's some, you can't maybe see very well on the projector, but there's some indication of a negative correlation between the breast cancer risk and the number of children, but the confidence intervals are really overlapping 0 here. The next thing we can do is remove any close relatives within our population, so it reduces the sample size, but it also potentially removes any genetic environmental confounding that can kind of inflate your heritability estimates. And upon doing that, we retained heritabilities of age at menarche and age at first birth, but you see that the number of children estimate decreases and is no longer significant. And when we try to estimate the heritability of our breast cancer risk here, basically it all disappears and the confidence intervals becomes really wide. You can also see the genetic correlations basically either fix at 1 or -1. I've cropped out the error bars here because they just became very, very wide. Okay, so our results do suggest that these genetic differences influence these two life history traits, the age at menarche, so age at development, and age at first birth in our population. But we really don't find any evidence for these genetic correlations between life history traits and this breast cancer risk. Can we say that this genetic variation plays no role? I would argue probably not yet. I think the error bars are really quite wide at the moment, and I think that it's probably just, the analysis is probably a little underpowered. There's also probably something in just the nature of trying to do these sorts of analyses on breast cancer. It's a very low prevalence disease, and it's essentially binary, so it's probably quite hard to leverage the variation in this trait. So this is very much an ongoing project. This is where I really need your ideas and help. So feel free to come up to me later or during the session. There are a few things I'm exploring now. First thing is to just try and bring up the sample size within the project. So we focused only on the most recent and highest quality genomic data within the Lifelines Biobank. But it's also possible that we can increase this using older waves, but it might reduce our number of SNPs available, but would certainly increase the number of individuals for which we have this genomic data. The other option is maybe just to try and explore some different sorts of genomic analyses. For example, kind of genomic time to event analysis. I mean, trying to take a more of a survival analysis approach, and I think that could be quite useful within this particular trait that we're dealing with. Or maybe also kind of genomic prediction scores or linkage to Sigma score regression. Okay, so that's basically all I had to say. I'd like to acknowledge my lab and all the support from my supervisors and collaborators, the ESEB that helped fund me to come here. And thanks you all for listening. And yeah, if you have any questions, please let me know. Yeah.
I have a question. I don't work on humans, but I was maybe wrong. But I was curious, you mentioned that breast cancer is a binary trait. Do you have information of when women were diagnosed?
Yeah.
Would that be correlated with their underlying risk? If you're younger when you're diagnosed, maybe you're more likely to have a higher risk.
Yeah. So I think the model that I ran, I also accounted for age as like a covariate, and yes, I think what you tell the model at that point is that you account for age, but broadly, women that have contracted breast cancer had a kind of higher overall predisposition to it. So I think that's what you kind of deal with there. But yeah, I think counting for age, but then of course it's you really plot it as like a linear variable, and maybe that's probably a bit unrealistic with so maybe trying to make that a bit more flexible, and I think maybe trying to explore these survival analyses are maybe the way to go. Hi, Anne. Thanks.
Hey, thanks very much for a great talk, great results. I was wondering, the public knowledge on this issue is very much influenced by, like, Angelina Jolie cases kind of things,
Yeah.
and I think it's she had like this BRCA
Yeah, the BRCA
mutation.
Yeah.
And so, did you find these candidate genes that are apparently very well known? And can you tell us how much genetic variation they explain? Basically, my question is, does it make sense to target only one mutation like this?
Yeah, so I think the BRCA gene is really this mutation that's really increases your breast cancer risk by 20-30%, I think it is. So it's prevalent. I'm not sure actually if it's in this population. I've not particularly looked for that, and I think a lot of the work that's done before has been trying to pick out these single genetic variants, and I think I'm trying to move away from that. But I do agree. I think at this point it maybe makes more sense to start maybe totaling up the effects of these different alleles, but I haven't looked at it yet. So.
Thank you.
I guess on like a similar thread, have you looked at the difference between maybe like triple negative or ER positive breast cancer?
Yeah, so I'd say a lot of the work I did on this so far is really kind of going through the pathology database and really trying to subtype them. So I was interested in this estrogen receptor positive breast cancer, which is the one that's really meant to show the sensitivity to your estrogen exposure. But at this moment, it's it's around 80% of cases is this ER positive case, and the triple negative is quite a minority, and I kind of wonder how much power we're going to have to look at that. But I think I'm also kind of exploring the phenotypic data of the questionnaires and trying to bring in some other variables, so I'm kind of wondering whether to maybe go down that road, but that's where I'm at. Okay. Right, thank you.